Good morning, everybody, and welcome to another summer immersion presentation. We're looking forward to what you've been doing all summer long. You are surrounded by friends, so be aware of that we're looking forward to what you have to share. Your mentors and friends are here, so looking forward. So our first presenter is Ana Martinez. Her presentation is Barriers to Latina Women Faced with the Prevention of Cervical Cancer. Ana? Looks good.
You guys can see this?
Yes.
Okay, perfect. Good morning, everyone. My name is Ana Martinez. Today, I will be sharing the research work I have been working on along with my RLC leader, Dr. Estela Vasquez. The title of my presentation is Barriers Latina Women Face with the Prevention of Cervical Cancer. In a woman's body, the cervix connects the vagina to the uterus. All women are at risk for cervical cancer, and it mainly occurs in those over the age of 30. The main cause of cervical cancer is a viral infection called human papillomavirus, most commonly known as HPV. HPV is passed from one person to another during sex, and at least half of active people will have HPV at some point in their lives. Screening tests and the HPV vaccine can help prevent cervical cancer. When detected early, it is highly treatable and associated with long survival and a good quality of life. One of the screening tests is called a pap smear test that is performed by a doctor and looks for precancers and any cell changes on the cervix that can become cancerous if not appropriately treated. Women are recommended to get their first pap smear test at the age of 21 and based on your results every 3 years after that. In the United States, cervical cancer used to be the leading cause of death for women, but in the past 40 years, the number of new cases and deaths have both significantly decreased. This is thankfully due to many women receiving regular pap smear tests. However, Latinas are disproportionately affected by cervical cancer when compared to non-Latina white women. In 2018, the rate of new cervical cancer cases per 100,000 women was highest for Hispanic women, which was at 9.3%. Compared to 7.4% for white women. The goal of this systematic review is to identify the barriers that Latina women face when it comes to cervical cancer screenings, as well as finding the most effective intervention and prevention methods. The review plan includes searching for articles in 2 databases, PubMed and Scopus. For consistency purposes, the terms uterine cervical neoplasms, prevention and control, and Hispanic Americans were used throughout all the databases. 702 articles were found on PubMed and 726 were found on Scopus. However, after adding the terms prevention and control and having removed duplicates, 259 articles remained. We are still in the middle of this review section and have only gone about halfway through, but so far 17 have been excluded. We composed a memo in which we are noting down what we have noted so far. At the beginning, there was no mention regarding HPV and its link to cervical cancer, and this was around the year 1970. Now that we are midway, there is more on HPV that emerged around the year 2005. There was very little on adolescent girls and their moms' reasoning as to why they would or wouldn't allow their daughters to get the HPV vaccine. One major thing we noted was that a lot of articles mentioned both cervical cancer and breast cancer with equal focus, meaning that these barriers are preventing women from not only getting screened for cervical cancer, but other cancers as well. California and Texas were the 2 biggest areas that most of this research was being conducted in. Results from the studies that we have so far reviewed identified barriers including communication, especially language barriers, transportation, and health insurance. Fear and embarrassment were also identified as reasons why women don't receive screenings. Some articles even mention a bit about healthcare providers, such as nurses and doctors, and how they should be prepared to educate their patients more on the importance of preventative cancer screenings. Some articles determined that community-based programs that involve women who go out and spread the word on pap smears and cervical cancer were more effective than using social media or flyers. This systematic review will be published in both English and Spanish, as well as in a very simple and organized format that is easy to read and understand for everyone, in order to promote the importance of cervical cancer screening behavior, especially among this rapidly expanding group of Latina women in the United States. Here's my works cited. And I want to give a special thanks to Dr. Estela Vasquez for guiding and helping me throughout this research process. And a huge shout out to NIH's Build Exit the Rice program for making all of this possible. Thank you, everyone.
Thank you. Great job. So, clap. This paper is now open for questions.
All right, this is Lisa. I have a question. Not quite a question. Anna, great job. That's beautiful. So as you're going through with your presentation, so heads up. So if you haven't connected with the community outreach and core community outreach research and engagement group for OHSU, so they are doing some of this work. And I imagine that your work and their work would be beautiful because they're trying to get a lot of these results out to the community, specifically working on these projects now. So if you have any interest, heads up, I would love to connect you if desired, but beautiful job.
Perfect. Thank you. I will do. Thank you.
All right. Any other questions? So Anna, how do you think what you have learned with?
Yes, go ahead.
Hi. So how do you think that what you have learned from doing this work in HPV can translate to COVID-19 vaccine hesitancy? And it's something that is contemporary. I don't know if the same reasons apply to both cases.
Yes.
You're the expert now.
Yeah, I think what I would say is that the main thing that all these articles were mentioning is that the importance about the HPV vaccine is to get information out there. A lot of the reasons why these moms were against getting the HPV vaccine for their daughters was because they didn't have enough information or they were too scared. So even just providing adequate information to them, and some of these articles were mentioning how to get the information out there as well. It was very beneficial, just like how with COVID vaccine, I would think, like as long as our parents are informed and everyone is informed with the right information, why wouldn't they get the vaccine?
Excellent, thank you. How much is, I don't want to monopolize. By the way, I have one paper on these topics, I'll share with you. But
Perfect,
how much, that's fine. How much is language an issue?
It's a very big issue because language is, your form of communication, and these patients are unable to establish a connection with their primary healthcare providers, which also that lack of a relationship doesn't motivate them to go to the clinics and get all their annual screenings done.
Excellent. We have one question on in the chat box. You mentioned one of the barriers being fear of being deceived. Can you say a little more about the fear and where you think that comes from?
Yes, I think it comes down because in order to get the pap smears, it's a very invasive procedure, so a lot of women are just having those gynecology visits, and especially if you don't know the language, if you don't have transportation there, they're just oh, so they kind of just avoid it. They're like, I doubt I'll even have it, so everyone has that mindset, and not a lot of women start going. Excellent.
Oh, here's the contact for the HPV project. Yeah, thank you.
Thank you.
We have a couple of seconds here. So I'm going to share something that we learned a long time ago when we're looking at HIV, and it was the role of, especially in the Latino community, of las abuelas, because what we learn is that the fathers are more hesitant than the mothers in terms of a vaccine to protect against a sexually transmitted condition. So we learned that the grandmothers could override the sons' unwillingness to allow their daughters to be vaccinated. And it's a tool. It's one more tool in terms of reducing the hesitancy from Latino families to allow their daughters to be vaccinated. Just something to think about, because they might play a role as hierarchy. In the hierarchy, they can override sometimes the fathers. All right, great, thank you. Looking forward. I'm glad we started with a topic that I had some familiarity. I'm equally nervous, by the way. You think that I'm not worried, but I am. Our next presenter is Shanice Sueda. She's mentored by Dr. Beth Habecker on chondroitin 4 sulfate proteoglycans in the prevention of sympathetic axon outgrowth. Take it away, Shanice.
Good morning, everyone. My name is Shanice, and I'm in Dr. Habeker's lab at OHSU. I'm excited to share about the research project that I've been insisting on, which is the conjoint 4-sulfate proteoglycan, preventing sympathetic axon outgrowth in the heart. If you look at here in the animation, when in the healthy heart, the sympathetic nerves are spread out evenly. However, when a heart attack or myocardial infarction occur, damage occurs to the nerves, which is the process of denervation. And then the cardiac scar is formed, as you can see here. Within the cardiac scar, it contains a combination of protein and sugar side chains known as conjoint sulfate proteoglycan, or CSPG. What CSPG does is it actually inhibit or prevent nerves from going back to the scar, resulting in arrhythmia and sudden cardiac arrest. Conjoint sulfate proteoglycans, or CSPG, actually can be further modified by sulfation. A study conducted by Pearson revealed that in the central nervous system, of sulfation that occurs in the fourth positions of sugar side chain of CSPG prevents nerves from going back into the scar after a spinal cord injury. This fourth position I'll be referring to as 4S-CSPG. And how it does is it activates a phosphatase known as PTP sigma. And the activation of PTP sigma actually inactivate a receptor known as tropomyosin receptor kinase A, or TRK-A. What TRK-A does is it actually promotes axon outgrowth by interacting with nerve growth factor, or NGF. So when PTP sigma is activated, it actually prevents TRK-A from binding to NGF and promote axon outgrowth into the scar. Other study had also shown that sulfatase B, or ARSB, removes sulfation of the fourth position of sugar side chain of CSPG, allowing axon to grow back into the scar. How is this relevant is we hypothesize that 4S-CSPG are present in the cardiac scar, and the removal of 4S-CSPG by using ARSB promotes axon regeneration in the cardiac scar. How we did is we collected a cardiac scar tissue by performing myocardial reperfusion surgery. These cardiac scars were actually then ran through a Western blot to actually determine if 4S-CSPG are present in the cardiac scar after an MI. Then we took a ganglia and allowed it to be co-cultured alongside with myocardium tissue, and some ganglia were actually treated with ARSB to see if ARSB modulate or remove the fourth position of CSPG. We use image J to actually measure the axon outgrowth. When we ran the Western blot from the myocardial reperfusion surgery, it was revealed that 4S-CSPG are highly upregulated or expressed in the myocardia after an MI. So here the stain is highly concentrated compared to the healthy cardiac tissue. I did not mention this in the previous slides, but CSPG is naturally existing proteoglycan. On the right of the Western blot, when we increases the ARSB concentration, it actually removes more of the 4S-CSPG. We use this Western blot to actually determine the concentration needed to actually remove the fourth position of CSPG. Here, I wanted to show the image of how it looked when the ganglia were co-cultured alongside with cardium tissue. So our definition of targeted tissue is defined here. When we actually measured using the image data, it was revealed that 4S-CSPG does inhibit the axon outgrowth towards the tissue, as it's shown here in the axon length. However, when we treat it with ARSB, it actually allows the axon to grow back into the similar length to the control group. So 4S-CSPG does allow
ARSB does allow axon outgrowth towards the cardiac scar tissue, hopefully prevents arrhythmia and cardiac arrest. In conclusion, 4S-CSPG are present in the cardiac scar after an MI, and it inhibit axon outgrowth towards the cardiac scar. And a removal of 4S-CSPG by using ARSB restore axon outgrowth towards the cardiac scar tissue. Thank you. I'd like to take this time to thank my graduate mentor, Matthew Blake, for guiding and supporting me through the research, and as well as the other lab member in Dr. Haberker's lab. I would also like to say thank you to the NIH Build Ex Vivo program and the faculties for giving me an opportunity. Thank you.
Great job, Shanice. Excellent.
Thank you.
So this paper is open for questions.
I got one.
Mm-hmm.
Shanice, what's been your favorite thing in the lab? So like you've done some Western blotting, you've done some surgeries, you know, like there's lots and lots of different like methods and things. Like, have you found like one of the things that you're like, I love that, or like a specific pathway? So like as it's getting really, you know, like specific in the molecular path, like what have you really loved and has stood out for you?
Yeah, so to be clear, I haven't performed the surgery. It was actually my other lab member that did the surgery. I'm not actually trained to do that, but I actually did enjoy running the Western blot, even though it does take a lot of time, and the process behind of like the reasoning behind of why things are done is actually more interesting. Hope that answers the question.
Excellent. We have a question in the chat box. Is it better for axons to grow into scar tissue?
So yes, we believe that when a denervation or when the sympathetic nerves are damaged, what happens is you're more susceptible to getting arrhythmia and a sudden cardiac arrest. When we allow the axon to actually grow back into the scar, it might prevent from a future cardiac arrest.
Excellent. So I didn't get this. This is a human model. These are not animal models. These are human models you're working on, right?
It is actually a mouse model.
A mouse. Okay.
Yes.
All right. Okay. How do you make a mouse have a heart attack?
So Melanie and Diana is actually the one that performs the myocardial reproduction surgery. Okay. And what they do is they tie a tube around, they tie a tube to actually mimic a heart attack, and they release it to have this like reaction. Sorry, I'm not.
No, that's fine. This is quite exciting. And other great comments here in the chat box about your visual. And there's a comment from Dr. Marriott. Go ahead, you can say it. She says, I've heard from spinal cord injury that nerves tend to not grow into scar tissue easily. One other question is there a reason for the upregulation of the protein in scar tissue?
So for the first questions, I'm not entirely sure how different it is compared to the spinal cord injury to a cardiac scar. It would actually depend, I'm assuming. I'll actually get back with you. I'll talk to my PI about it. For the second questions, there are enzymes that I haven't mentioned in the presentation that actually promote the sulfation of conjoint sulfate proteoglycan, or the CSPG. And this specific sulfation actually interact with a phosphatase known as PTP sigma and actually prevents that axon regeneration to occur.
Excellent.
Hope that answers the question.
Cool. Excellent. Lisa, there's a comment on the spinal cord injury also. Thank you, Matt. Thank you. Great presentation. I appreciate. The next presenter is Sophia Kim. She's mentored by Dr. Andrea Goforth, and her presentation is quantitative analysis of the reproducibility of the seeded stober method. Take it away, Sophia.
Good morning, everyone. My name is Sophia Kim, and I am the member of Dr. Goforth's lab at PSU. And today I would like to share my research project about the quantitative analysis of the reproducibility of seeded stober method for preparing silica nanoparticles. So first, what is nanoparticles? Nanoparticles are particles that has a size range of 1 to 100 nanometers. And today, various nanoparticles are used in many fields because they have a unique properties that compare to their bulk material, such as greater surface area, photoluminescence, and magnetic properties. And many of these physical and chemical properties of nanoparticles are known to be highly size dependent. So for our lab, we focus on silicon. Silicon dioxide nanoparticles are promising in biomedical and therapeutic applications due to their excellent biocompatibility, good thermostabilities, and low toxicity. An example of biomedical application of silica nanoparticle is drug delivery. Here's an image of hollow mesophorous silica nanoparticles. The hollow space inside the particle allows to load drugs and use for the drug delivery. And as mentioned, the physical and chemical properties of silica nanoparticles are also size dependent. So controlling the size of the silica nanoparticle is an essential part of the development of synthesis method. To synthesize science-controlled monodisperse silica nanoparticles, our lab has developed a new modified stober process called seeded stober method. This method uses small, highly monodispersed seed to grow larger silica nanoparticles. These larger silica nanoparticles are then used as a template to synthesize hollow mesophorous silica nanoparticles. As you can see, this pane represents a mesophorous silica coating. It forms a shell around the core, and as we remove the core, we will have a hollow mesophorous silica nanoparticles. And currently, our initial studies have shown that this method is effective in producing monodisperse and larger silica nanoparticles. However, we are still lacking statistical evidence to prove the reproducibility of the seeded method. The goal of my project is to establish an appropriate statistical method and evaluate the batch-to-batch reproducibility of the core part of this method. The size uniformity of the core is important as it directly affects the size uniformity of the final hollow mesophorous silica nanoparticles. The method of this research is divided into 3 big parts: synthesis of silica nanoparticles, sample collection and size measurement, and statistical analysis. First, the silica nanoparticles will be synthesized using the seeded stober method. Here we have 2 solutions, one in the round bottom flask with ethanol and TEOS. TEOS is the precursor to silicon dioxide, and we have the second solutions with water, ammonia solution, and the seed. When the first solution was mixed for 15 minutes, and after the solution with the seed will be added to the TEOS solution all at once. The reaction will proceed for overnight at room temperature, and thereafter it will go through the high-speed centrifuge and washing process to obtain the silica nanoparticles. And this process will be repeated for at least 3 times to test their reproducibility. The sample from each batch collected will be collected and measured using TEM and ImageJ. TEM is a transmission electron microscope which could generate highly magnified images. And here is a sample of TEM image obtained by our group member. And this image will be now analyzed using a software called ImageJ. ImageJ provides effective tools such as contrast and threshold to analyze the image and obtain the size of the particles. The collected data of the size measurement will be then used for statistical analysis, where the one-way analysis of variance, ANOVA, will be used to test any statistically significant difference between the mean diameters of the nanoparticles. This is a data from our group member, Lydia. It's a size measurement of mesophorous silica nanoparticles, and this is just a sample ANOVA run by me. So my hypothesis is that our synthesis method will provide increased synthetic control over beam diameters and size polydispersity of silica nanoparticles. And as a conclusion, with the further research, we are hoping to suggest a reliable laboratory method of silica nanoparticle synthesis, as well as a statistical treatment to nanoparticle reproducibility. So here are my references. And thank you, everyone. I would like to thank Excittle for giving me the opportunity to present today. And also thank you to my research mentor, Dr. Goldford. And special thanks to Lydia for providing the TEM image and the data. And thank you to all the Excittle staffs and advisors. I'll be happy to take any questions now.
Thank you, Sophia. Well done. Great job. All right. So this paper is now open for questions. Sophia, is your work has an application to a specific disease that you're thinking about?
There is not yet a specific disease I'm thinking about because this is focusing on the synthesis process itself. So we're like for my research project, it's not really considering the applications of the silica particles.
All right. So we have one, Dimitri, go ahead.
In the long run, Carlos, that we were going to make contrast agents out of them.
Okay, good. Thank you.
Hi, Sophia. Great job. I was just wondering, have you or Lydia looked at statistically looking at the thickness of the shells as well, or just the overall size?
So I haven't been working in the lab yet because I just came here like a few weeks ago. But for my focus, we're focusing on the synthesis part of the core. So for now, the thickness of the shell is not quite a part of my research.
Oh, okay. So you were still looking. Okay, you had a picture of the HMSNs, so I was a little confused. But you're just looking at the stober cores. Okay, that makes sense. Sorry.
There's a question on the chat box. What is your favorite application of how silica nanoparticles will be used in the future?
I think the drug delivery that I introduced in the presentation is very interesting because like that could be also applied for like cancer treatments and other chronic diseases because you can load drugs and you can deliver to that specific target and release the drug. I think that's very interesting.
Thank you. I just want to mention that we have some scholars who are URI scholars, and they are brand new to Éxito. They're doing summer immersion with summer induction, and their makes throughout the whole presentation. But this is technically their first summer, and we also realized that the pandemic didn't allow for everybody to be able to be 100% physically present in the labs. So we appreciate that you made progress under the circumstances. So thank you. Any other question for Sophia? All right. Great job, Sophia. Appreciate it.
Thank you.
Yeah, you're welcome. So our next presenter is Winnie Cadiz. Her mentor is Dr. David Wagner, and she will talk about the access to mental and behavioral health services among youth and novel interventions in children healthcare. Take it away, Winnie.
My name is Winnie Cadiz, and I'm a senior at Portland State University studying psychology. And today I will be presenting on access to mental and behavioral health services among youth, novel interventions in children's healthcare, also known as NICH. So let's start off by introducing NICH. NICH is a program that helps youth with chronic or complex illnesses. The model on the left encompasses the components of the program that address the known limitations of standard health interventions. An example of this would be interventionists going outside of the usual business hours or going to a patient's home for a check-in. What we know about NICH is that it's associated with less frequent complications. As you can see in the graph on the left, there is a decrease in emergency department visits and admissions for youth in NICH during NICH, while we also know that NICH is associated with a reduction in disparities. As you can see in the graph on the right, there is an increase in appointment attendance for the BIPOC population. However, what we don't know is how access to mental and behavioral health services differs between multiple chronic illnesses among youth in NICH and a comparable group. So for my research project, I aim to assess the engagement in mental and behavioral health services for youth with type 1 diabetes, cystic fibrosis, and chronic pain in NICH compared to similarly referred youth. So the sample included youth with these 3 diagnoses who were referred to NICH due to high social risk. 72 youth received NICH, and 134 youth in our control group were referred to NICH but denied by insurance. So we collected electronic health record data for 3 years, and with that data, we coded it to reflect mental and behavioral health appointments. Then we calculated the mean appointment attendance for each time point for each diagnosis. And on the pie graphs on the right, you can see a breakdown of race and ethnicity for the NICH and control group. As you can see, they're quite similar, but the control group being more diverse. So our initial findings were that there were differences in appointment averages according to each diagnosis within 3-year time period. And in general, the chronic pain patients had the most behavioral health appointment attendance throughout the 3 years, as you can see on the graph on the right. Here, I broke down the average appointment attendance for each diagnosis. And we ran an SPSS analysis as well for each diagnosis, as well as across groups. And we found that there was no significant difference in terms of percent prior to referral. Here in this graph, you can see that there's a slight decrease in appointment attendance for the NICH population, while the control group remains stable. And we also found that there was no significant differences in terms of a number of kids that access the mental and behavioral health services over the 3-year time period. So we can conclude that NICH youth did not have an increase in mental behavioral health appointment attendance. And there were no meaningful differences between the NICH and the control group in their appointment attendance, which is the reason is unclear. However, we can infer that it may be due to the fact that NICH youth have weekly behavioral health services at home, which isn't reflected in electronic health record data. And because this is unclear, we can say that further research must be done and must be conducted on mental and behavioral health outcomes in NICH. Lastly, I want to talk about limitations. Our electronic health record did not reflect all the possible mental and behavioral health appointments. We only collected OHSU appointments, and the inpatient data were not included. Also, another limitation is that our sample is quite small and racially and ethnically homogeneous, so it's not really generalizable. However, future studies could definitely look at outpatient data or referrals, which would be super interesting. Finally, I'd like to acknowledge that this research was made possible through the Leona M. and Harry B. Helmsley Charitable Trust and Build Exito. I would like to give a special thanks to my PI, Dr. David Wagner, and the rest of my RLC. And that will conclude my presentation. Thank you all for your attention. Any questions?
Thank you. Great job. Great job. All right. So this paper is now open for questions.
I have a question. First of all, great job, really interesting. I'm curious if you think that if there would be reasons that the control group was denied by insurance for the services that might have impacted your results. So maybe there were some factors in that group that made them seem less like they would need NICH, and therefore, you didn't really see a big difference. Does that make sense? Do you think there's anything like that going on?
I'm actually unsure about how the referrals work and how our insurance, whether they take NICH or not. I can ask my PI who's here, and he can speak on it if he wants to, and I can get back to you, but I'm not sure about the whole insurance process. Thank you for your question.
Yeah,
it's a great question. Certain insurers won't cover the program, period. So it is a confound that the folks in the control group are from different insurance companies than those who are in the NICH group.
Got it. Okay, thank you.
All
I have a
right.
quick
Yeah, go for
question.
it.
It's
Uh-huh.
really just like sort of along the lines of some previous questions. Like, what was it that really stood out to you as something that you would want to like follow up and research about this program, or what has been sort of like one of those threads as you realize that like some of your research was inconclusive? Like, what would you love to go forward with?
There are so many things I want to do with our population, especially with our incoming studies. We can see like a larger sample, and what was most interesting was that the general increase, or not, how much appointments that chronic pain population had. I really would love to look more into them and maybe compare them to other diagnoses.
There's a question on the chat box. Did no-shows have a big impact on findings?
I think all of the data that had zeros in it had a big impact on our findings, as well as the youth who did have a lot of appointments. And it was really hard to measure the averages because of those large skews. So it could have, but we can't know for sure.
Mm-hmm.
Thank you.
So, clearly this has implications for patient care, but it probably has implications for cost. Do you know more or less if this is something that will reduce costs if it's implemented with high affinity?
Great question. Yes, it NICH. I think we looked at how much money NICH saves hospitals and whatnot. And sorry, I was reading the chat. But yes, the NICH can definitely be cost effective.
Okay. How do you, you're reading the question, so I could read it. My understanding is that niche population is quite diverse. How do you think that impacted the finding? Was the control group equally diverse? Actually, I had a question about the control groups. Were they I didn't hear the word randomization, right? Were they randomized or just matched?
No, they were not randomized. And I think NICH is quite diverse for Portland, for Oregon, for sure. And I think that definitely has an impact on the findings. Was the control group equally diverse? No, the control group was had a few more races and ethnicities in it than the control group.
And Ms. Miller asked whether NICH model being used or tested for other populations who need better access to mental health services. Yeah.
It's just a general musing.
Yeah. But I think it has implications for multiple different populations in different settings. Yeah, it's great, very exciting. So thank you, Winnie. Appreciate it. Great job. Yeah. All right. So our next presenter is Thejanee Liyanaarachchi. I did practice. Liyanaarachchi, and forgive me if I misspell it or mispronounce it. And so she is going to talk about mutant viruses from hell, Mutegenizing the cleavage site. She is mentored by Dr. Ken Steadman, famous TV celebrity.
Hi, everyone. My name is Thejanee. I work at the Extreme Virus Lab at Portland State University, and this is Mutant Viruses from Hell, Mutegenizing the proteolytic cleavage site in VP1. So as you can probably tell, I work with viruses, and the most basic definition of a virus that you can find is it's a nucleic acid, so DNA, RNA, wrapped in a protein coat called a capsid. Sorry. And if you look at these pictures, you can see most of the viruses that we know of are of these helical, spherical, and complex structures. However, the ones that our lab studies look nothing like that. Ours look more like these lemon or spindle-shaped viruses seen in picture C. So these viruses are called SSVs, sulfolobus spindle-shaped viruses. These viruses are found worldwide in hot springs. So these hot springs are both thermophilic, so hot, and acidic, so it has a pH about 3. So essentially it's boiling orange juice. And like all viruses, it has a host. It's an archaeal host called Saccharobacillus sulfatericus. And you might be wondering, why are we studying viruses that live in hot springs if it doesn't impact our day-to-day lives? The reason that these specific viruses of interest is because while we are made up of the same macromolecules, so proteins, DNA, etc., we cannot survive these environments. But how could the same subunits of proteins and DNA be able to survive in these environments? So essentially, we're figuring out what structural differences exist or how these structures are made that makes it compatible with environments that the rest of the organisms cannot. So we're going to use SSV1, so sulfolobus spindle-shaped 1, as our model. And because we're looking at the interaction of the virus to its environment, we're predominantly studying the capsid structure, so that protein coat. So the lemon-shaped capsid is actually made from 2 proteins, virus protein 1 and virus protein 3. We focus on virus protein 1 because it's the major subunit in the capsid. And as virus protein 1 is quite a large protein, so we couldn't necessarily mutagenize and test everything. So we actually did a comparison of 10 SSVs seen here. And then if you see this E66 position right here is highly conserved, and it's actually conserved in many of the 40 SSVs. So we decided to focus on that specific site. This site is of importance to the protein because it's a proteolytic cleavage site. It means that when the amino acids are strung together, it's actually cut at this location before the protein can function as the protein's intended. Previous research has actually mutated that E, or glutamic acid, to an alanine or glutamine, other amino acids. And this resulted in infectious viruses, but they didn't have that lemon-shaped structure. So my research is actually continuing on them. So my goal is to actually mutate that position to the other amino acids. So the way that we did this, our methods, is we took a vector that has the wild-type genome and we broke it up. And then we took the segment that has the position that we're looking for and we mutated that specific position to the other 19 amino acids. Essentially, we put it all back together, amplified it in E. coli, and eventually put it into the host so we can get virus production, and then test if it's infective, and then test the structure through TEM, which is transmission electron microscope. So as of today, we have 14 amino acid changes, but we have 44 mutants. This is because there's more than one sequence for each amino acid. And if you see on the other side, we have some intermediate results. So this first gel actually shows the fragments when we cut the fragments up to make sure that they were the right size. The plates with the little white dots are actually individual mutants. We were not able to test all of it. Those circles show the ones that we tested. This last gel actually shows the some of the gels that we tested before we sent them in for sequencing. These latter first 2 are actually methionine mutants. And you can actually see an image that we actually took of one mutant, the methionine mutant, right here. And as you see, the shape is actually like the lemon-shaped structure. So moving forward, what I need to do is actually infect the remaining mutants to the host and check if it's infectious. And the way that we do that is through a halo assay. And this is an example of a halo assay that was done in my lab previously. And then check the structure of these viruses through TEM. But as you remember, we only have 14 of the 19 amino acids. So we have to actually produce and examine the remaining mutants. So that's going forward. I would like to acknowledge Bill Dixito, PSU, Dr. Sedman, my PI, Dr. De La Higuera, my mentor, and Denise, Jono, and Jenny, who were members of my lab that helped me quite a bit. My bibliography. Thank you.
Excellent. Great work, Thejanee. Great work. So this paper is now open for questions. So go ahead.
Well, I'm curious. So you're going to get asked about viruses all the time. So we're hearing about with climate change and all of the ice starting to melt, and there's lots of viruses that are starting to come out of the woodwork, for example, or come out of the ice technically. But so as we're kind of moving forward with, some of those things, like what are things that, either you're looking out for or that others should be looking out for? I know that you're focusing on, really, heat friendly viruses, but, these will be others. So any thoughts on that?
I mean, not specifically, but I mean, something that would be interesting to see is how these maybe archaic or ancient viruses actually impact like modern day hosts, because they may not necessarily impact systems in the same way that it did back then.
Thanks, Thejanee.
Thejanee, this is a out of this world question, literally. So there is a possibility that this type of viruses might exist in other planets. But is that conceivable? I know we haven't been there, but what do you think?
I'm not sure about viruses themselves, because we're removing the ability of reproducing in those environments, because viruses require a host. But I think if we have like maybe I guess kind of like RNA-based stuff, RNA-esque viruses that can like self-catalyze, maybe it's possible. But especially if it's talking about heat and stuff, so micromolecules can definitely withstand those conditions. So it is possible.
Cool. So, and evolutionarily speaking, how old are these viruses that we can say, they've been around for millions of years? This is a relatively new virus.
The specific one that I'm studying?
Yes.
The reason that we're not sure about its actual age is because we think it's ancient simply because it's found worldwide. It's found from the Philippines to the US to Iceland to Russia, and it's highly, highly conserved. Its genomes are highly, highly conserved. So that means there had to have the most simplest explanation, which means that they came from the same ancestor, which I guess could refer to how ancient it is. But the question is up for debate, because it's actually studying evolutions of viruses is really difficult because of its mutation rate.
Yeah, thank you. Excellent. Any other question? I know we've Lisa and I have been monopolizing the conversation. What's your favorite virus?
I guess my favorite virus, not because of like studying it or anything, but because it's kind of where I got.
You have to be careful what you say. Dr. Stedman is very sensitive about picking.
I think because I got my interest in virology came from it was actually dengue or dengue in my culture, especially because it's a huge issue from where I come from. So it's kind of where my interest in viruses started. So I would say it's my favorite simply because it led me to start studying or be interested in virology.
Excellent. Thank you. By the way, the CDC dengue laboratory. Oh, there you go. There's a picture. The CDC dengue laboratory is in San Juan, Puerto Rico. So you can do your PhD there. All right. Excellent. Excellent. Appreciate. Thank you, Dr. Stedman. So our next presenter is mentored by Dr. Alan Teo, Jeanette Doorenbos. She's going to talk about the understanding risk factors, assessment and interventions for social withdrawal, a systematic review of Hikikomori. Take it away, Jeanette.
Thank you. Can you hear me all right?
Yes, sounds good. Perfect. Excellent.
Okay. So, hi, I'm Jeanette Doorenbos. I am a senior at PSU pursuing my bachelor's of science in psychology. I work in Alan Teo's lab doing a systematic review of Hikikomori. So you're probably wondering, what is Hikikomori? Hikikomori, defined by the Japanese Ministry of Health, Labor, and Welfare, is the condition where a person suffers from severe social isolation, a lifestyle centered at home, and refusal of work, school, and hobbies for a duration of 6 months or longer. What we currently know about Hikikomori is that there is no official diagnosis of this condition, and the term originates in Japan, but studies have recently shown a global presence and that it's more common in young men. Our key questions focus on 3 subjects: the risk factors, mostly characteristics associated with Hikikomori, the assessment tools used for diagnosis and measuring prevalence, and lastly, interventions and their outcomes. Here we are focusing on the interventions used in Hikikomori, while some studies still exist about that. Our systematic review followed the steps presented in a Prisma flow diagram. Our first step was the identification and literature search step, where we looked at 5 databases listed here to pull for potential literature review. Each database was recorded as such, with each step and the number of results listed for tracking purposes. Our total number of articles was initially 3,373, but after deduplication, we ended up with 1,775 articles in total. Our next step after that was the screening and stage 1 process, that allowed us to look at all the abstracts and titles of the articles to dwindle down the amount of literature before full text review. We use the inclusion and exclusion criteria listed here to code articles as 1 for keep or 0 for discard. After screening, we were left with 103 articles. We are currently here at the eligibility and stage 2 stage review. This is where we review the full text of the 103 articles using a written flowchart-like document shown here. As we review the article, we answer the questions and either continue to the next one or code according to the questions. For example, if one of our articles was not in English or Japanese, we would answer no here and code X1 in our coding sheet. We would end up with approximately 20 articles after this process. Our last stage before writing the systematic review and our next step is data synthesis, where we use thematic analysis to identify patterns in the included text in terms of how they relate to our key questions. In this stage, we will pay attention to the consistency of the data and the degree they're presented in. This will influence how they're presented in our final paper. Like most systematic reviews, we are interested in presenting the most up-to-date knowledge about Hikikomori in relation to its risks, assessment, and interventions. We hope to help define what is known about Hikikomori, inform people on the condition, and ask the question, what's next for the future directions of research, assessment, and interventions for Hikikomori? I'd like to give a special thanks to my principal investigator, Alan Teo, my study team, Olivia Lashley, Tiana Wu, and Jenny Park, and Build Exito. Any questions?
Excellent. Great job. Great job. So this paper is open for questions. So I would imagine with COVID-19 and lockdowns that this is a prime subject to study, given how we have involuntarily been asked to have many of the Hikikomori risk factors. Has COVID-19 exacerbated the number of people with this condition?
So COVID-19 has played a big part in our literature review so far, or our systematic review, because out of the 1,775 articles, I can say that a very high percentage of those had to do with COVID-19. And that's because a lot of them mention social isolation, but do not focus on the aspect of social isolation itself. As for like the occurrence of Hikikomori, we can't for sure say that COVID-19 would increase the amount of the occurrence of Hikikomori because it's a temporary isolation. And while it has gone on for longer than the duration of what is expected from Hikikomori, it's not a voluntary social isolation. It's more of a, you have to do this.
I kind of like this social isolation. Now maybe I'm, yeah. So there's one question from Dr. Gofor.
Yeah, interesting presentation. I had watched a YouTube video about a computer game programmer who is How do you say it? Anyway,
Hikikomori.
yes, thank you. Wonder like how this term is used culturally. Do people call themselves Hikikomori, or are other people calling them this? I get the sense it's not a clinical thing.
So it's not a clinical thing yet. Hikikomori is a Japanese-based term, and it was originally thought to be a cultural bound syndrome. So it was supposed to be something that only occurred in Japan, but we found that it actually occurred in more places around the world, and that culturally there's a bunch of names for Hikikomori and how it occurs. Like there's the agoraphobia, which is the fear of going outside, which could classify as Hikikomori. And so, you know, it's not the official diagnosis or the term, but it still exists everywhere.
Interesting. And is there like status assigned to it, either positive or negative? Like it's good to be one or it's bad to be one?
Definitely in the literature, it seems, and also like the culture around it seems very bad. Japan has kind of a focus on combating Hikikomori because most people who are suffering from Hikikomori are young men who are going to enter the workforce. And so when they become Hikikomori, they withdraw from everything and they aren't helping their community or anything. They aren't doing anything for the community. And they're also usually residing in their family's home. So they're also kind of becoming I wouldn't say like a burden, but they are there and they are just like in a room.
Yeah.
So it's an exciting subject. Is there a mutually exclusive differentiation between being introvert and extrovert? I mean, can you be extrovert and yet have Hikikomori, or this is something that primarily happens among people who are introvert?
I'm sure that could be a study on its own. Though I think that people who are more introverted would be more inclined to fall into Hikikomori tendencies. Usually what we've seen with the systematic review so far is that it has to do a lot with conditions that also associate with depression and anxiety. And there's been less about personality traits so far. I've seen a lot about asthma though, and relating to Hikikomori.
Interesting.
What about correlations to economic conditions? Like if there's a perceived good job market versus bad?
Yeah, I believe there was some stigma around, at least specifically in Japan, that the hopelessness with having to work so much and having to be the sole provider kind of put that pressure to fall into Hikikomori onto the young men. And so there is definitely that pressure, but I wouldn't say it's because in Japan specifically that because there's a bad job market, but more because they are expected to work.
All right, we have one last question. We can go on here. Is there a connection between MCAD, mast cell activation disorder?
I could not tell you. If my PI is still in here, he might be able to get some insight into that, but I have not seen anything in the literature so far.
I think I saw Dr. Teo in the group, but
yeah, I'm here. I don't want to interrupt. I think Jeanette's been doing a stupendous job handling the wonderful questions. So yeah, there's a little bit of biologic research that's going on. No direct connection. There is some work at sort of like correlation between neuroinflammation and social isolation withdrawal. So it's not my area of expertise in particular, but there is some biologic research sort of in that area going on.
Excellent. Wow. Excellent topic. So we need to move on, but if you have any additional questions, please send it to Jeanette and Dr. Teo. So thank you. So our next presenter is Shanthia Espinosa. She is mentored by Dr. Lisa Marriott. She's going to talk about exploring Native Hawaiian Pacific Islander students' motivation in STEM within higher education. Take it away, Shanthia.
Howdy and hello everyone. I am Shanthia from Saipan, Northern Mariana Islands. I am Chamorro, mixed race, and I am majoring in public health, community health promotion. And I'm doing my research under my amazing PI, Dr. Lisa Marriott, and lab at OHSU School of Public Health. I'm so glad to be presenting my research here in Portland. I just moved here 3 weeks ago from Hawaii, and I'm super excited to share my talk with you and on exploring Native Hawaiian Pacific Islander students, NHPI and STEM. So provided here is a map of the Pacific Islands and the grouping of each categories, which I will break down briefly on the next slide. But as we can see here, the three main categories of North and South Pacific Islands are Micronesia, Polynesia, and Melanesia. Here's another visual that I adapted from ASCO. And here we see Melanesia, which is known for the islands of Black or melanated skin, Polynesia known for its large island populations, and Micronesia, you guessed it, small islands. So overall, Pacific Islands consist of small and large diverse populations, and each populations have their own unique characteristics. So a little background on NHPI and STEM is that there is limited research on NHPI communities. And in addition, research on Pacific Islanders was often misinterpreted, and an example is shown on the graph above in orange. And by disaggregating data of API, this led to a lack of representation of Pacific Islanders. And in 1997, the census introduced the NHPI category shown on the table below. So now we have our own category. And previously, I had the honor to conduct archival research with the McNair Scholars Program on NHPI motivational factors and opportunities. And for my research today, I will be implementing the Real D standards, which I will explain briefly on the next slide. And as a public health major and researcher, equity and inclusion is a big part of community success, and I encourage you all to look into the Real D standards. So in addition to the introduction, one of the common themes for my research with McNair is that NHPI students come from low SES and are usually the first in their families to attain their degrees. And the Real D stands for race, ethnicity, language, and disability, encouraging inclusivity. And for this research, we hope to fill the gaps by identifying strategies for supporting student motivation in STEM, and by implementing the Real D example shown on the slide. It goes into depth on each race and ethnicity. Will allow us to aggregate data of NHPI across other research topic areas. And for methods, we will be recruiting NHPI STEM students across Oregon, Washington, Hawaii, American Samoa, NMI, and Guam. And we will be conducting 10-minute surveys with the Portland Metro STEM Partnership. And I have an example of the questions here. One question is, I find topics related to science interesting, and that would be like positively scored. And then we have, I often feel bored in science, which will be negatively scored or reverse scored. And for feedback questions, we have, what is one piece of advice for staying motivated that you'd like to share with other STEM students? And in addition, we also have virtual focus groups, which will be 90 minutes. They are semi-structured, and we will be assessing strategies for STEM motivation and success. And this will be open to NHPI and other race and ethnicities who would like to participate. And for our research schedule, here's a quick breakdown. As you can see, we plan to recruit in October, which is very soon, as well as our virtual focus groups, which we plan to complete in November. We also plan to conduct our qualitative data analysis from November to December. And by next year, we hope to generate e-feedback. And if you are interested in participating, email us at start@OHSU if you're interested, and we'll be getting back to you. And for our anticipated research findings, we hope to identify effective strategies for encouraging student success and hope that our findings using the Real D will expand data not only for NHPI, but other race and ethnicities as well. And to conclude, I would love to say Suzus Ma'asi and thank you to Build Exito, my lovely faculty mentor, Dr. Lisa Marriott, my amazing lab team, and everyone present here for our talk, and Build Exito for giving me this amazing opportunity. Thank you.
Excellent. All right, this paper is open for questions.
I have a question. Way to go. That was awesome. Yay. I'm curious about if you have some outreach strategies that you're going to use in October to reach people.
Ooh, we will most likely have some flyers. We're on Qualtrics, so we have like this QR code that we can do and send it out in emails. What else? I would just say emails and flyers. Yeah, and just word of mouth too. I'm in McNair and we're in other programs, so it'd be nice to have our lab share. But if any of you are interested, feel free to email start@ohsu.edu. Yeah, and I will get back to you. We'll get back to you. Thank you.
Do you have a target sample size, and then how would you make sure that you have enough representation from Polynesia, Melanesia, and Micronesia?
That's a great question. In regards to our surveys, I feel like the more participants we have, the better. And then for our virtual focus groups, we do plan to have about maybe 10 to 12 from NHPI communities and an additional 10 to 12 for other race and ethnicities, just so we can have that kind of pool. And yeah, I'm not too sure if I answered your question, but yeah, I hope that we could get like a really diverse group of students. That's my hope.
Yeah, no, that's cool. Has this ever been done?
That's a great question. I'm not sure, but I would love to know if anyone has done it and connect with them if you ever find out.
Yeah, I mean, that's great. I mean, the fact, even if somebody has done it, we don't know much about it. So it's going to have a huge impact. Great contribution. All right. Excellent. Thank you. Good job. So our next presenter from the lab of Dr. Lisa Marriott, the effect of Sin Lek Rice on cognitive function, dietary patterns, and fecal microbiota of elementary school children in Chiang Rai, Thailand, by Angie Gee Setthavongsack.
So, Sawadika and hello everyone. My name is Angie Setthavongsack and I am a part of Dr. Lisa Marriott's lab. And today I am doing a project called The Effects of Sin Lek Rice on Cognitive Function, Dietary Patterns, and Fecal Microbiota of Elementary School Children in Chiang Rai, Thailand. So what is Sin Lek rice? So rice is often harvested in September to October. This is located in Chiang Rai, Thailand, and this rice is often called rice against diabetes. It contains a low glycemic index. It also hinders the absorption of sugar into the blood, therefore making it suitable for patients with diabetes. It also improves functioning of the pancreatic system. You can see that the colors between the Sin Lek rice and the white rice is very different, and that's because it's a crossbreed between Hom Nin and Thai Hom Mali rice, Hom meaning fragrant. So how does food affect your brain? So this is an example of the gut-brain communications. Up above, there's the vagus nerve. The vagus nerve brings information from the inner organs, such as the gut and liver, to the brain. And the bottom shows the circulation and communications of the immune pathways and neural pathways to the brain as well. Here's something a little more in depth about the gut-brain axis. And this includes the gastrointestinal system, the nervous system, also the immune system, also relating to obesity, type 2 diabetes, and the inflammatory bowel diseases, as well as neurodegeneration, causing multiple sclerosis, Alzheimer's disease, and Parkinson's disease. So the objectives of this project is to determine differences in the microbiome pre and post Sin Lek Rice. Second, to quantify changes in body mass index pre and post Sin Lek Rice. And lastly, specifically for my portion of the project, is to estimate the effects of Sin Lek Rice versus traditional white rice on cognitive function as determined by performance on short-term working memory and attention tasks. So here are some photos of the newly renovated kitchen in Chiang Rai. This is all new pots and pans and typically what a rural village looks like as well. Here are some photos of the children taking the OHSU surveys as well. So here is the timeline. We started collecting data around December of 2019 and started the diet intervention on December 11th of 2019. This is when grades 1 through 3 had the placebo effect, only eating white rice, and grades 4 through 6 had a 1-to-1 ratio of Sin Lek and white rice. And then at T15, so week 15, that is when we did the washout period, and that is also when COVID-19 hit. So there was a little bit of skewed times with this part, but this is where nothing is happening. And then back on track at T56, that is also when we swapped diets. So that is when grades 4 through 6 had white rice, while grades 1 through 3 had Sin Lek Rice. All right, so this is the methodology of Me Pha Leung University. I can't go into too much detail about this section, but they did do, they collected data through fecal sample matters and did DNA analysis with the bacteria there. As for OHSU, we did 3 different types of tests. So the impulsivity scale included terms such as I plan tests carefully, I do things without thinking, and I don't pay attention, ranging from never to always. Second, we had the Corsi block test, which is a sequent test kind of game for the children to click on, memorize, and click on the dots in the specific order. Last, we had a visual attention game where a red dot would appear on the screen and they needed to click it as fast as possible. So our results. So to the right, you have the microbiome results from Me Pha Leung, and at the bottom, you have the bifidobacteria, which had a significant change from T0 to T71 at the end of the experiment. I also can't go into too much detail about the findings of Me Pha Leung. However, we noticed that this was the most significant out of the bacteria in this graph. To the left is the cognitive and diet results at baseline for OHSU, and we noticed that at baseline, females had better memory than males, including leaning towards high sugar and high salts. So in conclusion, we're still doing data analysis and we're planning on doing furthering at T0, T52, and T71 as well. I'm also working with the Fulbright Association and Dr. CM at Me Pha Leung to hopefully extend this project onto mental health, and as the gut also relates to depression and anxiety. So I'd like to thank my lab. Tila's not in here, but thank you, Tila, as well. NIH Build Exeter, OHSU, Me Pha Leung, Portland State, and Fulbright. Do I have any questions? Thank you.
Excellent, excellent. By the way, before the end of your presentation, I have already ordered a bag of Sin Lek Rice. It's on Amazon. It's on its way. I should get it tomorrow.
Oh, how exciting. Let us know how it tastes.
Yeah, I look forward to it. So you already had an impact with your research.
Thank you.
Any other questions? Any other comments? So there's Sin Lek being patented. Is there a patent for it, or is that a naturally occurring product? Is it genetically modified? I don't know if this is part of the question, but maybe you can comment on all this topic. Yeah,
that's an interesting question. I actually don't know whether or not it's patented or not, or if it's crossbred specifically. I assume it's made by by the villagers there and also like scientifically like approved like that. But I can't be certain. Oh, developed. Thank you, Lisa.
So I'm going to check the rice when it comes tomorrow, if it's genetically modified or not. So I'll have an answer. Earlier you mentioned that it has a hypoglycemic impact or hypoinsulinemic impact.
Low glycemic index.
Okay. That's yeah.
I am unsure exactly what that does, but I assume it's all right.
No, it's better for you. I mean, we do know
Okay.
that white rice, and there's some epidemiological study that do show that there are high rates of diabetes among populations that consume high amounts of white rice.
Yeah.
Cool. So I want to make sure that somebody asks can ask questions. Can you discuss a little bit more about the importance of the bacteria that was highlighted?
Thank you for the question, Tila. So I can't go into much detail about bacteria as that's not part of my specific part of the research, but from what I do know, different bacteria definitely affects the inflammation in your intestines. And obviously there's different bacteria that is more affected with high sugar than high salt. So I can only assume that some growth in bacteria will either be really good or really bad. And we're still in the research of that. And so I'm sure Dr. CM also knows a little bit more. And so I could totally ask him for you and get back to you Monday.
So, I mean, at least you did find there is a significant difference in one of the bacteria. You did mention there was a significant difference by gender in one of the items. Is that, yeah, I mean, that's not the effect of
so
eating rice. Yeah, go ahead.
This is at T0, so this is at the beginning, and we noticed that females did have better memory and leaning towards high sugar and salts. We're not too sure of how that will affect further in the study as we're still researching that, but it's something that I'm definitely interested in learning about as well in the future.
Okay, excellent, exciting.
Thank you.
Looking forward. I'll let you know how the rice tastes. Thank you.
Sounds good. Thank you.
All right. So our next presenter is the real Kai Kubi, mentored by Dr. Bruce Goldberg, identifying increase of needs of pre-COVID-19 in comparison to post-COVID-19.
Okay. Hello, everyone. My name is Daryl Kubi. I'm currently doing my RSC at the lab of Dr. Bruce Goldberg at OHSU. And today I will be presenting my project on how the novel coronavirus impacted the needs of Medicare and Medicaid patients pre-COVID to now. To start, I have a little background on what I've been studying the past year in this lab. About a third of Americans have unmet social needs, potentially resulting in health effects and high healthcare expenses. Improper structural components that impact how individuals live are the determinants of health that are to blame for the health disparities that disadvantaged populations suffer. These health inequities have become even more pronounced in the light of the current novel coronavirus, which has predominantly impacted low-income areas. So with this observation, the Centers for Medicare and Medicaid Services have developed a 5-year study that tests whether systematically detecting and treating the health-related social needs of Medicare and Medicaid participants by screening referrals and community navigation services would affect the cost of healthcare and usage. This multidisciplinary collaboration among community-based organizations, human services providers, and clinical care providers will test health-related social needs interventions of varying intensity. In the consortium's 28 county areas, the consortium's CTOs offer care for Medicare and Medicaid clients, It was created to thoroughly screen the populations for social needs connected to health and to provide navigation assistance. And with this, we asked, how has the novel coronavirus impacted the needs of Medicare and Medicaid patients? This initiative is organized into 3 tracks, each of which is based on potential services delivery methods. However, the AHU program has been using tracks 2 and 3. Track 2 revolves around assistance, where AHU provides community services navigation services to assist high-risk beneficiaries with accessing services. And currently, the AHU program is now on track 3 alignment, where AHU encourages partner alignment to ensure that community services are available and responsive to the needs of beneficiaries, such as screening and providing resources, identifying health-related social needs, and reducing barriers. So our target population revolves around most clinical sites that provide care for Medicaid and Medicare patients of all ages, including primary care, behavioral care, dental emergency department, labor and delivery, public health, and mobile clinics. To answer our question, the study is conducted thoroughly through systematic screening, which can take place in either clinical or not clinical setting, and can be done through secured email, phone call, or a text. The survey is about a 5 to 10 minute survey to see whether the patient has any health-related social needs. We then determine whether that is a low risk or a high risk, and provide community resources. In addition, high-risk patients can get a call from the 211 patient navigator to help them access those resources. With our result, if we look at the 2 graphs of needs of Medicaid and Medicare population, as of August 2021, we have screened a total number of 15,613 patients. As you can see in the chart, it shows that the health-related social needs of these 2 populations have increased compared to before COVID-19 started. Lastly, to assess the impact of the pandemic, the data collected after March 11, 2021, when COVID-19 was declared a pandemic to now, we found that there is a significant increase in emergency department visits among Medicare and Medicaid patients. Currently, AHU is improving communications and coordinations between the health and social sectors, which might make it easier to form partnerships to meet social needs. Also, to commit and addressing social comorbidities. This research was supported by the NIH Build Exito, OHSU, Orprin, Bruce Goldberg, Ann King, Zoe Major, and Rosalia Goto. There's my references, questions.
Good job, Daryl. And very timely, given where we are circumstances today. So thank you. So this paper is open for questions.
I have a question. I'm wondering what other areas you expect to see health impacts related to isolation, if there are areas that have shown up more.
Oh yeah, totally. So we currently screen whether their social needs, such as transportation, what I mentioned earlier, and social isolation and safety. So
Daryl, I know this is where you were placed. It's the Office of Rural Research Network. But did you notice any difference between rural, very rural, semi-urban, urban areas? Did that have an impact?
After doing that, or to answer your question, like more rural like places really don't have has a low impact, like compared to like more towards the city where everything is more expensive.
Yeah.
Yeah.
So this basically happens across the board, not just in very rural areas.
Yeah, it's all over the board, especially low-income neighborhoods.
All right. Any other questions? Good job, Daryl. Appreciate. And our last presenter, Randa Hanna. She is mentored by Dr. Kristen Makiewicz-Seget. And the topic is COVID-19 pandemic-related stress and its association with nutritional intake during pregnancy. Take it away, Randa.
Hello, everyone. My name is Rhonda, and the research I am working on involves COVID-19 pandemic-related stress and how it is associated with nutritional intake, specifically the three main macronutrients, carbs, fats, and protein during pregnancy. So a little bit of background. What we know about stress is that it can be acute or chronic. Acute stress is short-term stress, while chronic is long-term. Chronic stress can have serious and long-lasting negative health impacts. Stress may affect individuals differently, and it can express itself as physical symptoms, which come in varying degrees. For example, some people experience headaches, dizziness, insomnia, or muscle tension. However, stress can also have a huge impact on mental health and brain plasticity. This is especially crucial when it comes to the pregnant population, because the COVID-19 pandemic has been lethal to at-risk populations, meaning there is increased risk for the mother and for complications in the fetal environment. How HPA axis plays a role in stress response. So the hypothalamus pituitary adrenal axis plays a major role in the body's fight or flight response. When an individual is stressed, the hypothalamus will release corticotropin hormones, which will bind to the pituitary adrenal gland, and that releases adrenocorticotropin hormone, or ACTH. The ACTH will then bind to the adrenal cortex and release cortisol. Cortisol will then initiate a negative feedback effect, and it will allow the body to quickly respond to the stress and return to its normal state. Nutrition can help improve HPA axis response to stress. However, when individuals are stressed, they tend to burn carbs faster because of increased insulin levels caused by cortisol release. They also tend to make unhealthy eating decisions, which typically consists of a lot of sugar and fat. So this is what we are looking at. The aim of this study is to describe the relationship between COVID-19 pandemic-related stress and macronutrient intake during pregnancy, and whether it differs for individuals with lower levels of pre-COVID-19 perceived stress. We predict that there will be a relationship between pandemic-related stress and macronutrient intake. Specifically, we hypothesize greater pandemic-related stress will be associated with greater levels of fat and carbohydrate intake. The main determinants of HPA axis activity are genetic background, early life environment, and current life stress. This is the reasoning behind why we decided to look at early life adversity as well, which brings us to the second part of the hypothesis. We predict there will be a stronger relationship between pandemic-related stress and carbohydrate and fat intake in individuals with lower levels of early life adversity, because the HPA axis is not overactive in those individuals. After we identify that a relationship exists, we will then assess mindfulness as a moderator, because mindfulness has been shown to reduce stress responding in pregnant individuals at both a behavioral and biological level. So in this study, participants complete an ASA 24 food diary at baseline and 34-week gestational age. Macronutrients, so carbs, fats, and proteins, intake for participants will be extracted from the ASA 24 and examined to see if a relationship exists between these macronutrients and pandemic stress. Mindfulness as a potential moderator for stress and eating habits will also be examined through a dispositional measure called the 5-facet mindfulness questionnaire. And it's repeated at multiple time points during pregnancy. This questionnaire assesses things like awareness of the actions and allowing negative feelings and thoughts to pass without much contemplation. To assess early life adversity, we will be looking at measures that examine adverse childhood experiences, for instance, the childhood trauma questionnaire. So here is an example of what the ASA 24 data looks like. It shows when it was completed, types of foods consumed, caloric intake, protein intake, fat intake. It'll also show carb intake and many other variables. Future directions. So we hope to bring awareness to the impact of an environmental stress on the prenatal system specifically. And we hope to bring awareness to how individuals with early life adversity may be at an altered risk. We would like to disseminate this information to susceptible communities and inform them about potential non-pharmacological and non-therapeutic interventions like diet. I would like to acknowledge the NIH Build Exito and the Build Exito faculty for support, and my PI and RLC mentor, Dr. Kristen McCoy-Xvieri. These are my references. Thank you.
Excellent. Great job, Randa. All right, this paper is open for questions.
I have a question. Wonderful work. And sorry if you mentioned this and I missed it, but do you have thoughts on whether or not mindfulness, that sort of moderation might look different depending on their early childhood trauma situation? Like, do you have any expectations about how that might play out differently depending on sort of where they are on that spectrum of trauma to none?
Yeah, so I think so mindfulness effects, so it can be a very therapeutic thing, especially at people with ACEs or like adverse childhood experiences or early life adversity. They may already know some mindfulness practices to help them deal with the previous trauma. So, but also there could be some that do not have that coping mechanism or they don't practice mindfulness. So I think it will be different.
There's a comment chat in the chat box about research examining epigenetics and methylation of stress receptor among those with trauma. And then there was a study of prenatal nutritional impacts done after the Dutch famine that followed the progeny into adulthood. How long does your study plan to collect data?
Yeah, so our data is going to be collected during the COVID-19 pandemic. So it's just how long the pandemic lasts, but we will be collecting it at 34-week gestational age and baseline.
Excellent. Are you collecting any biomarker data or any other, I want to call it more objective, but some other clinical measures that are rather stable? And I have some ideas. I know one faculty in the School of Public Health who was interested in looking at heart rate variability in relation to how people eat and mindfulness. It's as a marker of how you're being able to control the anxious eating is to be able to look at heart rate variability. It used to be, I'm answering the question, but it used to be somewhat somewhat difficult, but with the new phones that actually they do measure heart rate variability. I don't know if this is something that you guys are doing or there's any other biomarker that you didn't talk about.
We are not collecting data on heart rate, but we might be looking at exercise or physical activity later on and assessing it like in a similar way as we will be assessing intake. Yeah, so that might be future work.
Yeah, that's fine. Good. All right. Any other questions? All right. So we have basically come to, that's Dr. Claire Wheeler, Lisa. So I think you're our last presenter. Is anybody, did I miss, anybody wants to present or haven't presented? This is the last one, right? Yeah, okay. So great job, everybody. I mean, this has been extremely satisfactory. Everybody presented. You guys took the time you needed to do. You answered questions. Amazing. You're ready for graduate school. However traumatic you think this 5 minutes, probably the longest 5 minutes of your life, but you did great. So we're really proud of you. Want to also thank the mentors for getting you to where you are today and preparing you and anybody and everybody who was behind your presentation today. We look forward to your future. I know this is the foundation to more, hopefully more building upon what you have done so far. So we're really proud, really excited of the work you have done. We're going to have another set of presentations on tomorrow. Yeah, Thursday. All right, Thursday. So and feel free to come over and support your classmates. Any other departing comments? Any other questions overall? All right, so looking forward to seeing you next, and again, congratulations, well done. So now I think we now have lunch, right? No, it's not lunch.